All right, hi guys. So this video is going to be the first lecture of our new genetics unit. And with this genetics unit, first of all, I do feel like this is probably one of the most interesting for you guys. You guys always have like tons of questions about how genetics works, and you tell me about funny situations about the colors of your eyeballs or your family, your cousin's eyeballs or whatever. But one thing that's going to look a little bit different is this genetics unit is going to have a lot of stuff that to me feels a little bit mathy, like you're going to actually have to write stuff down. So for those of you at home, my little virtual learners, again, I sent a packet of papers home to you. You probably won't get it for a few days, which is just fine. But because as things progress through the unit and things get more difficult is when you're going to really need it. All of you are going to have virtual notes. So everyone is going to still be using a virtual notes packet. All of you, if you look on my screen here under genetics unit resources, the first thing posted should be the genetics notes packet. And it's going to look really similar, like we've done so many times together now. And you can see we're going to open it up and start here today with genetics notes. Most of today's lecture and actually the next few days are really going to be review. A lot of this you'll have seen, you should have already seen in middle school. Okay, so that's the notes packet. That's where you need to be following along. All right. So again, right now, all of you are just following along with the virtual notes packet and making sure to pause as you need to fill in the blanks. So when we talk about genetics, genetics is just the study of heredity. And heredity is what is so interesting to like almost all of us about passing traits from parent to offspring. So far you should have, again, in middle school, now I'm going to start from the beginning, so don't freak out, but in middle school, you should have already done some practice with monohybrid Punnett squares here on the left. And remember, mono means one. So this is a Punnett square looking at just one trait. So it may be the color of a flower or the length of the stem or whatever it may be, looking at one trait. We're going to practice monohybrid squares here for a few days. And a lot of you sitting in class tell me, oh my God, I'm so bored. I've done this 1,000 times. And that's great. But we will be progressing to a lot of stuff that you've probably never seen. We'll spend a day or two on these big giant dihybrid Punnett squares. There's 16 squares in this Punnett square. The beauty of a dihybrid Punnett square here on the right is that we're actually going to track two traits together. And we'll see if two individuals potentially have offspring, what are the chances that their offspring would have these two traits. So we'll slowly progress into dihybrid Punnett squares. And then I think one of the coolest parts of this unit is looking at pedigrees. And this is basically like a really nerdy family tree. So with pedigrees, we'll spend a few days looking at pedigrees, starting from the very beginning in case you've never seen them before. But pedigrees are just really tracking a big old family tree, but tracking like one particular trait or one particular disease. So most often pedigrees are done in order to not so much track eye color, right? We don't really medically care about eye color, but we're thinking, like a genetic disorder or genetic disease or something like that. So we can't talk genetics without talking about old Gregor Mendel. So Gregor Mendel is known as the father of genetics because he was the first scientist to discover the law of genetics. So there's a picture of Gregor Mendel. He conducted many experiments on pea plants, like peas, like the peas that you would eat. And that his work on pea plants really provided the foundation for heredity. And I'm gonna slowly just give you a little tiny background about why he used peas and what he was looking for with the peas, for example. So it is, you guys, estimated that Mendel cultivated and tested over 28,000 pea plants, like thousands of pea plants, okay? And what he realized was that the plant's offspring retained traits from the parents, right? So he was able to like look at a specific pea plant, record observations about the plant, and intentionally breed plants, and then look at their offspring. And so he was really the first person to notice that traits are passed from the parent plants to the offspring. Here's one example of where he did his work. So those are some examples of pea plants. He was, sorry, I thought I had a slide about it, but he was a monk, and monks live in, you guys always tell me this. What do I do without my 30 students telling me where monks live? Monasteries, right? So, and why did he choose peas? Well, first of all, they can be grown in a small area. So as you saw in that picture, that wasn't like a huge, massive plot. So they can be grown in a small area. One particular plant can produce lots of offspring. So even if you're like growing peas to eat, like one plant will have lots of peas on there. They produce what we call pure plants when allowed to self-pollinate, and we'll kind of talk about that in just a sec. But they can also then artificially be cross-pollinated. So in this picture here, he has what looks like a tiny little paintbrush, and he's actually cross-pollinating. So he's like brushing one flower to get the little pollen, and then he goes and brushes another flower to pollinate the second flower. Just a reminder that angiosperms are anything that have flowers, and so anything that have flowers, but you guys don't realize how many things actually flower. Every fruit flowered, right? So oranges were a flower, peas were a flower, maple trees flower. So lots of plants flower, and in order to reproduce with flower, there's male and female parts. And so if you ever have seasonal allergies and pollen in the air makes you sneeze, you are literally sneezing because of plant sperm. Okay, so most pollen, if you Google image it, it's amazing. Pollen looks like really spiky circles. And I don't have allergies. My husband has terrible allergies and he gets sneezy all the time. And so basically, if you imagine those really spiky circles in the air, almost pretty microscopic, getting sucked into your nasal passageway, you can imagine why it makes you sneeze. And that truly is a plant's sperm. Self-fertilization is when one flower, like in the image here, so here we have a flower, it's producing pollen, and then it also has an ovary. So the pollen are here on the stamen, and if you can picture, like if you've ever put your face in a flower and sniffed it and then you get pollen on your nose, that's the pollen, and that's the male sperm. And then the flower also has a female part. So they have the long pistil here, and deep down in the little green part that you would almost not even really notice is actually the female ovary. The male pollen needs to make its way down the pistil to fertilize the ovary. And that one fertilized ovary is what ultimately is going to grow your apple, for example. So self-fertilization is where the same flower can just fertilize itself. Cross-fertilization is when pollen actually drifts through the air, probably makes a lot of people sneezy in the meantime, and makes its way to another flower. A lot of plants are allowed to become fertilized in both of these ways. So they can become self-fertilized, but also cross-fertilized. Okay, so what all started this was Mendel began working with his pea plants, and he produced what we call a pure strain by allowing a plant to self-pollinate. Okay, so its own pollen fertilized its own ovary. And we call that a pure strain. He also then very strategically began to cross-pollinate. So he would say, if I take this pure strain over here and this pure strain over here and cross them, what traits are passed on to their offspring? And in order to do that in a really controlled manner, and this still, botanists are still doing this today, they take what you can even see in the picture in the top right here. They literally take a little thing, a paintbrush, and they kind of take the paintbrush and kind of tickle the stigma there with the pollen, and then tickle the other flower in order to pollinate it, right? So they tickle one flower and tickle the other. This is exactly naturally what bees do, right? So bees go from one flower to another and they pollinate flowers along the way. But again, he did this in a really controlled manner. I'll also say that botanists out there, if they're trying to do this in a controlled manner, they don't want the pollen to just be able to blow throughout the atmosphere, right? So they actually put little plastic bags over the flowers to literally not allow any contamination or allow the flower to be fertilized by pollen from the air, right? So they want to be able to do this scientific study and kind of control what is pollinating what. Okay, so when we, when we look at genetics here, we're going to be looking at traits. And the term trait refers to any characteristic that can be passed from parent to offspring. So anything that's passed from parent to offspring. An example of this, the classic one I always use is eye color. Okay. Within every trait, though, you guys, there's lots of options, right? One of the forms of a trait is what we call an allele. So you may have an allele for brown eyes, or you may have an allele for blue eyes. Depending on the trait, there may be literally like one set of alleles. You got one from mom and one from dad. Or we'll learn in this unit actually how heredity is actually most commonly like really complicated. And when it comes down to it, you guys, you didn't really just get one allele for eye color from mom and one from dad. And that's why we have such a wide range of eye color, in skin color, you guys, and height. All those things where we see like a huge range is actually because it's not near as simple as we're going to see genetics here in the next few days. Okay. Some examples of traits and alleles that Gregor Mendel was working with. So an example of a trait was like the seed shape, and then the alleles are the options. So for seed shape, he was looking at round or wrinkled seeds. For seed color, he was looking at yellow or green. For pod shape, he was looking at smooth or wrinkled, so on and so on and so on. So you get the idea that there's a trait, and then the alleles are, think of the alleles as like the possibilities. Those alleles can be what we call dominant. And the dominant allele is the variation of the gene that will produce a certain phenotype. So even in the presence of other genes, this dominant allele will be presented. So dominant alleles are always present and they're always expressed. I'm sorry, if they're present, they're always expressed. Dominant alleles are represented by a capital letter. We'll use, it doesn't matter what letter you use along the way in the Punnett square. We'll talk about it as we do some practices as well. So an example here of the dominant allele would be capital B. The other option for alleles can be recessive. So recessive is the variation of a gene that will not produce a phenotype in the presence of a dominant allele. So I've heard some people call the recessive like the weaker of the two, and it's, don't think of it as the weaker. It's just the recessive. So in order for the recessive trait to be expressed, you have to have two recessive alleles. So this is only expressed if two copies are present, and it's represented by a lowercase letter. In this case, I've been using B's, so this would be a lowercase b. Okay. When geneticists kind of, I mean, even beginning with Gregor Mendel, and they're tracking generations, it's really important that we're able to name the generations and understand what these names mean. So the generation that we start with is called the P generation, or the parent generation. They breed and then produce the F1, or the first generation. Okay, so F1 stands for the first generation, and it was produced from the P generations mating together, right? Mating. Then we take the F1 generation and breed them together and end up with the F2 generation, or the second generation. So we have the P, the F1, and the F2 generations. So here's an example of the P generation. So remember, Gregor Mendel always started with true or purebred plants. So we have a purple flowered plant here, and he's going to cross it with a white flower. And then their offspring are what we're going to call the F1 generation. And then we take two of those and breed them together and look at their offspring. And so this is exactly what really initially caught his attention, was what in the world? How can we take a purple flower and a white flower and breed them, and all of their offspring are purple? But then if you take two purple offspring and breed them, white flowers pop up again. So if you look here, it's odd until you take a high school genetics course. It's odd that that the white flower now reappears, right? So how did it skip a generation? And that's what you're going to understand. Okay, so we'll be looking at a couple things. We'll be looking at genotypes, which is the actual gene combination. Examples of a genotype are like the two alleles. So big P, big P, little p, or little p, little p. The phenotype is the physical characteristic expressed. So phenotype, think physical characteristic. So if you look at the pictures up here, here's an example of a big P flower, and another big P, and a big P, little p flower, and then a little p, little p. So the genotypes are those alleles I just said. The phenotype, though, would be describing the physical characteristics. So in this case, big P is purple. Big P, little p would be purple. And then little p is white. So the phenotype here is either purple or white flowers. This kind of looks when you first glance at it, it looks complicated, but I love it because it tracks the generations. So here we have a purple flower. We start here on the left side. This is our first Punnett square. We're starting with the P generation, and we start with a purple and we start with a white flower, and they have babies. And remember, all their babies, this is the F1 generation. And remember, we said that they were all purple. Well, if we take two of the F1 generations and breed them together, so over here we now have the two purple flowers. And in this case, if you look at the Punnett square, you guys, you can see that all of their offspring are big P, little p. So that's what we've have here in the second cross. So we have big P, little p, and big P, little p. And then we cross them with a Punnett square, and this is how we see that the white flower is now going to pop back up. Okay, so this white individual here got a little p from each of those parents, and it was masked by the dominant allele, big P. Okay, sorry, I'm trying to keep track of time. Oh, for heaven's sake, I didn't start it. Okay, genotypes, when we look at, remember the alleles, they can be homozygous, which means that this genotype has the same alleles. So homo means the same. Examples of homozygous are like big P, or little p, little p. Okay, so they're the same, either big or little. We may also call homozygous genotypes pure or purebred, and you'll see that. And then heterozygous is a genotype with different alleles. So hetero means different. The only possibility with heterozygous, always, 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 is a big and a little, because they have to be different, and those are only options. So big P and little p. And examples of this, or sorry, we also call this hybrid. So you'll see, I'm going to throw those words at you as we move along. I'll maybe call it a hybrid individual, and you'll have to know that that individual then would be big, little. Okay, so homo or hetero. So big P here is going to be homozygous. Big P, little p is heterozygous because it's one big and one little. Heading down here to the white flower, little p, that would be homozygous because they're the same. And then big P, little p here again is going to be heterozygous because it has two different alleles. Okay, so we're going to kind of put this all together, and we're going to take a look at this funny little cloudberries. This is an example of an herbaceous plant found in boreal forests. And when we look at these cloudberry plants, there are two options for colors. So interestingly, there can be red berries or there can be yellow berries. The red berries are dominant and the yellow berries are recessive. So in this instance, the trait we're looking at is the berry color. The alleles are red or yellow. Those are the two options. And because red is dominant, I'm going to give it a big R. And then yellow berries, I'm going to give a lowercase r. So the possible, the possible combinations here, the possible genotypes are big R, big R, little r, or little r, little r. When we look at the phenotypes of those individuals, because big R is dominant, any individual that even has one big R is going to have the dominant trait. So big R is going to be a red individual. Big R, little r is going to be a red individual because it has one dominant trait. And then the only way to have little yellow berries is to have two of those recessive traits. So little r is going to give you a yellow berry. Okay, so here we are using a Punnett square. And I like this because it puts together the sperm and the egg. So essentially, I want you to imagine that every egg has one allele to offer. So here we have the eggs at the top, and every sperm has one allele to offer. So here we have the egg alleles and the sperm allele options, and each individual and their offspring just get one allele from each parent. So for example, this individual in the Punnett square got a big A from mom and a big A from dad. So big A, big A. This individual got a big A from dad and a little a from mom, and so on. Okay, so let's take a look here. When we're looking at Punnett squares, you guys are going to be given the parent genotypes. Okay, so in this instance, the mother's genotype is big A, little a, and the father's genotype is also big A, little a. Notice that the mother's alleles are kept together. Okay, so here's mom, big A, little a, and dad is on the side. It does not matter where you put mom or dad, as long as you keep them together. Okay, so as long as you put mom on top and dad on the side, or you could put mom on top and dad on the side. It doesn't matter as long as you just keep their alleles together. And then in a square here with 4 squares, we're going to call this is a monohybrid Punnett square. You're going to just carry down the alleles. So you're going to carry down a big A here and a big A here. And you'll carry a big A here and a little a here. and a big A here and a little a, and then this individual will get a little a from each parent. Notice that I have made sure to put the dominant allele first. So for example, the heterozygous individuals here are big A, little a. It won't necessarily be wrong if you don't do it that way, if you don't put the dominant allele first, but your life is going to be so much easier when we do the huge dihybrid squares with 16 squares and 4 alleles in every one. That's a lot of letters. And I swear it's easier if you just get in the habit right away to put the dominant allele first. Okay. So here we go. We're going to do a little practice. So we're going to look at the eye color is the trait, and then the alleles are brown or blue. And I tell you that brown is dominant because I show you with the big B. So brown is dominant and blue is recessive. I have the parents filled in. So the father's on the top here with big B, little b, and then the mother's on the side with little b, little b. Go through and we'll fill in the squares. So as you can see, the first square here gets a big B and a little b. Notice I put the dominant one first. This square is going to get a little b and a little b, so little b, little b. This square is going to get a big B carried down and then a little b carried over. And then another little b and little b. So, sorry. When we look at the offspring then, and remember this is just a prediction. So when we look at the offspring, we're going to predict that 50% of their offspring here are going to be big B, little b, and 50% of their offspring will be little b, little b. Little b will be blue eyes because they have 2 recessive alleles. And then the heterozygous individuals are going to have brown eyes because they have at least 1 dominant allele. Okay, so we're still looking at eye color, and brown is dominant and blue is recessive. So what if a homozygous brown-eyed man marries a heterozygous woman? So homozygous brown-eyed, we know that he must be big B because he has brown eyes and it's homozygous, so he has to have the same alleles. Heterozygous woman is going to be big B, little b, so she's going to have 1 of each type of allele. So there we go. I filled in the square for the parents. So here's the dad, big B, and then the mom is heterozygous, big B, little b. I'm going to go ahead and fill in the squares. So I'm going to carry my alleles. I'm going to get a big B and a big B, and a big B and a big B, and a big B down and a little b, and then a big B and a little b. So all of their offspring are, the phenotype of all of their offspring are brown eyes because they all have at least 1 dominant allele. And then the genotypes, however, are going to be big B, and big B, little b. Okay, so for the first time, and again, so those of you in the classroom, you're going to have a packet that looks like this, which is the genetics practice set. I've never done this before, but I think it'll work really nice. So this is for you that are in the classroom. Those of you that are at home, this is on its way to you, but go ahead and pause it if you need and just get a piece of scrap paper. These first Punnett squares are going to be simple enough that you'll be just fine. But I do encourage you to actually get some scrap paper and follow along. So this is going to be separate from your notes packet. This is literally just practice problems that we'll do together in every notes, like every lecture. So today we're going to do just the first page together. And this is just so you guys have like a handwritten reference to go to. And I intentionally tried to put 3 really good practice problems that'll set you up for success with the practice that you guys will do alone. So here we go. We're going to look at question number 1. Oh, Goodness. Where's my marker? Okay. So in your practice set packet, for those of you that have it, we're going to do number 1. So number 1 says, in one species of dung beetle, shiny black big B is dominant over red little b. A heterozygous black male falls in love with a red female, and they start a family in a pile of herbivore dung. What will their offspring look like? So first of all, you're going to draw a Punnett square. Actually, sorry, yours has it. So if we look at our Punnett square, I said a heterozygous black male. Heterozygous, we know that he has to have 2 different alleles. So big B, little b. And falls in love with a red female. The only way to have a red phenotype is little b, little b. Okay. Sorry. Let's go ahead and fill in our square. So this individual is going to be big B, little b, and little b, little b, big B, little b, and little, little. So then when it asks what are the genotypes of the offspring, the genotypes would be big B, little b, and little b, little b. So if you look here, the only options of their offspring are to be either big B, little b, or little b, little b. And then if we look at the phenotype or the physical characteristic of their offspring, remember dominant is black, recessive is red. So we would anticipate that about 50% of their offspring are black and 50% red. So these individuals would be red. So we would expect them to be about half and half. If you want to listen to that again, pause and rewind. You can rewind all you want. Okay, let's move on to question 2. Dung beetles have spurs on the back of their back legs that can be long, big L, or short, little l. A homozygous long-spurred male meets a homozygous short-spurred female hanging around a fresh dung pile. Predict the genotype and phenotype of their offspring. So again, I'm going to make my Punnett square. And I intentionally chose the letter L. So some letters are harder to use in Punnett squares than others, and you just have to be smart about it. So with L's, first of all, homozygous long-spurred male. So homozygous long is dominant. So I'm going to give him big L, big L. And then I actually make it even more obvious. So the female short spurt homozygous short is, and I actually do like cursive L's, like little l, little l, make my L's look really different. So I'm going to go ahead and fill in my Punnett square. So I'm going to put a big L, little l. And you can see the genotype of all of their offspring are going to be heterozygous. So no matter if these individuals had 300 babies, they would all be heterozygous. So the genotype of their offspring is big L, little l. And then you can see sometimes I'll ask the percentage of the offspring. Sometimes I'll put it like you can see here, the phenotypes of their offspring, I put it out of 4. So all of their individuals would be heterozygous and also have the dominant phenotype, which is long. So I'm going to say 4 out of 4 would be long and 0 out of 4 short. Also, let me tell you guys, as you kind of jot this down, in these problems, zeros are just as important as another number. So telling me that 0 out of 4 are short is just as important as telling me 4 out of 4, okay? A lot of you, because it's 0, leave it blank. But don't do that. So 0 is just as important and is just as notable as any other whole number, okay? Any other number here. Okay. So that's 2. I think we have 1 more looking at these old dung beetles. All right, so question 3. After dung beetles mate, the female lays an egg in a dung ball that they created together, and then she coats the dung ball in fresh dung, saliva, and her own feces. Thick saliva, big T, is dominant over thin saliva, little t. A hybrid female with thick saliva mates with a heterozygous male with thick saliva. So what are the chances that they'll have a baby with thin saliva? So I'm going to do my square here. Okay. A hybrid female. Hybrid, remember, is the same as heterozygous. So she's going to be big T, little t. And I make my little t's look really weird. Some of your t's, you can't tell if they're big or little, you guys. Be really intentional about your letters, okay? So I have a big T and then a funny little t. So she's hybrid, mates with a heterozygous male. That's the same, right? So he's going to also be big T, little t. Let's go ahead and fill in our cross. Should look like that. The genotypes of the offspring, we actually have 3 options now. So the genotypes, big T, big T, big T, little t, or little t, little t. The phenotypes of the offspring, actually some of them will be thick saliva and some of them will be thin. So the phenotype here would be thick and thin. And then we want to know the chances of thin saliva. I actually can't see that. Sorry. Oh yeah, okay. So the chances of thin saliva. So the percentage. So if you think of it as quarters, right? So 4 quarters make $1. So 1 quarter here, this is the only one that's going to have thin saliva. And think of it as a quarter. It's going to be 25%. Okay. So 25% of the offspring will have thin saliva, or 1 out of 4. You'll just have to read these word questions closely and see what I'm looking for. So you'll have to watch whether I want the genotype, the phenotype, the percentage, or the fraction. And I will try to make it really obvious, I promise. I'm not trying to be tricky. I'm trying to set you up for the air test when I don't know what they're going to give you. And I think that's all for today. Yeah, that is all for today. Again, please go back, rewind if you need to watch some of these basic monohybrid crosses again. But now you should get started on, you're going to have a little hedgehog genetics worksheet. All right.